All right, let's talk about a shift in science that is, well, it's monumental. Think about this. What happens when you take CRISPR, our sharpest genetic tool, and you give it a super intelligent brain? That's exactly what we're diving into today. This is CRISPR 2.0, where AI is taking gene editing from just being revolutionary to something that feels like it's bending reality itself. Now, to really get where we're going, we gotta look back at where we started. CRISPR, it was a total game changer, and it still is. It gave scientists the power to go into the DNA of living things and precisely change the code. This completely transformed research and opened the door to therapies for diseases we once thought were totally untreatable.
So what does this actually mean? It means we've literally given the world's sharpest genetic scissors a super intelligent co-pilot. That is the core idea of CRISPR 2.0. We're talking about an AI that can predict, design, and discover things way beyond what a human could. And believe me, as we're about to see, this changes, well, pretty much everything.
Okay, so before we plug in the AI, let's do a super quick refresher. What exactly are these genetic scissors we keep talking about? The easiest way to think about CRISPR is like a word processor, but for the code of life itself. So it uses this thing called a guide RNA to find a very specific sentence in the massive book that is the genome. And then an enzyme, like the famous Cas9, swoops in and makes the change. It can cut, it can copy, it can paste, it can even just rewrite a single letter of DNA. And this right here shows just how much it has evolved. So the original tool, nuclease editing, it really did work like a pair of scissors, making a complete cut right across the DNA. But then the technology got way more sophisticated. We got base and prime editing. Now think of these less like scissors and more like the find and replace function in a Word doc. They can swap out a single letter of DNA without that big disruptive break. And that, of course, makes them much safer and way more versatile for fixing genetic diseases. But here's the thing. As powerful as this toolkit was, scientists started hitting a wall. For all its brilliance, CRISPR 1.0 wasn't perfect. Not by a long shot. There were some pretty serious challenges holding it back. So what was the holdup? What was the actual problem? Why did this revolutionary tool even need an upgrade in the first place? Well, it really boiled down to a few key hurdles. For one, the outcomes could be pretty unpredictable. You'd tell the editor to make a specific change, but what you got back wasn't always what you expected. And then there was the really scary one, the risk of off-target edits. The scissors cutting in the completely wrong place. Yikes. And maybe the biggest bottleneck was just that designing a new edit was slow and really, really expensive. Finding the perfect guide RNA and the best editor for a new genetic target, it was all trial and error. And in medicine, trial and error costs a ton of time and money.
And this is where our story takes a hard turn. To solve these incredibly complex, data-heavy problems, scientists turn to AI. This is the super upgrade. This is the exact moment CRISPR gets its co-pilot and becomes what we're calling CRISPR 2.0.
And look, we're not talking about some kind of sci-fi sentient AI here. We're talking about machine learning and deep learning. What that means is we can feed these AI models enormous amounts of biological data. We're talking millions of experiments. And by sifting through all of that, they start to learn the subtle, complex rules of gene editing. They spot patterns that no human ever could. Yeah, this visual here perfectly illustrates how it works. You feed the AI millions upon millions of data points, DNA sequences, the 3D structures of proteins, the results from tons of past experiments. It just crunches all that information and starts to build a predictive model of how biology actually works. It's basically learning the language of life so it can start making incredibly smart suggestions. So what does that look like in practice? Well, AI's role really boils down to three huge game-changing jobs. Job number one, it can predict the absolute best guide RNA to use for any edit. This maximizes your chances of success and, crucially, minimizes those dangerous off-target effects. Job two, it helps us engineer better, more efficient editor proteins, sometimes even brand new ones with new abilities we've never seen before. And job three, which is just mind-blowing, it can discover totally new editing tools that are just hidden in the vastness of nature's genetic code. The difference here is just staggering. It's a real before and after moment. Before AI, finding new tools was like trying to find a specific book in a library the size of the planet, but you only know the title. It was slow, manual work. But now, with AI, we can search for the shape of the proteins, their 3D structure. It's like having a 3D map of the entire library. You know exactly what you're looking for. And get this, instead of just tweaking existing proteins, which was super laborious, generative AI can now design brand new ones from scratch, perfectly optimized for a specific job. And this visual really gives you a sense of the sheer scale we're talking about. On one side, you have AI algorithms just plowing through these massive databases with billions of proteins from all kinds of microbes hunting for new natural tools. But on the other side, AI is going a step further. It's not just finding tools, it's inventing them. It's generating the code for completely new enzymes that have never, ever existed in nature. Okay, so what does this all actually mean for us, for the future of medicine? This is where we get a little glimpse of the incredible future that CRISPR 2.0 is actively building right now.
This right here, this is the new workflow, the new pipeline for creating a genetic cure. So you start with a mutation that causes a disease. Then AI helps you pick the absolute best tool for the job. Maybe it's prime editing. Then AI designs the perfect guide RNA for pinpoint accuracy. It can even help engineer the best way to deliver that editor into the right cells in the body. Literally every single step is guided, optimized, and supercharged by AI.
And why does this matter so much? What's the payoff? Because a tool like prime editing, when it's supercharged by AI, could theoretically correct over 90% of known genetic mutations that cause disease. Just let that sink in for a second. We're talking about diseases like cystic fibrosis, sickle cell anemia, Huntington's. This AI-CRISPR partnership is putting us on a path to fix them at their absolute source, the code itself. But maybe the most mind-bending part of all of this is what comes next. This quote here hints at what is kind of the holy grail, being able to test genetic cures on a computer before you even step foot in a lab. It's basically a flight simulator for gene editing. This is the next frontier, creating what scientists are calling virtual cells. By training AI on all this biological data, we can literally build digital twins of human cells right on a computer. This means we can ask the AI, hey, what will happen if we make this specific edit to this gene? And it will run a simulation predicting the outcome with incredible accuracy, all before a single real cell is touched. This alone could speed up the development of new cures by, maybe an order of magnitude? It's insane. And all of this brings us to one final big question. We've seen how AI has tackled CRISPR's biggest hurdles, making it more precise, more powerful, and faster than we ever thought possible. It's turned a revolutionary tool into an intelligent system for rewriting the code of life. So with AI as its co-pilot, maybe the question isn't what can CRISPR do anymore? Maybe the real question is there anything it can't?